We now move our attention to disaccharide, and later we will look into polysaccharide. Basically, disaccharide is a combination of two monosaccharide coming together by the effect of a glycosidic bond. And then you also have, in terms of the glycosidic bond, to be of variation, either beta 1,4 glycosidic bond, could be also alpha 1,4 glycosidic bond, could be also alpha 1,2 glycosidic bond. So there is variation to it. And then you have also, in terms of to differentiate between the monosaccharides, the numbering could be in prime or just simply 1, 2, 3. The above paragraph just gives you the explanation on the numbering, that is with prime or without prime, within the disaccharide, the monosaccharides. And then we will look into each one of the important disaccharide, that is maltose, lactose, as well as sucrose. So here you have the maltose being explained. So maltose is basically made up of two alpha glucoses coming together by alpha 1,4 glycosidic bond. And it is found in starch that has been hydrolyzed by either enzyme of the amylase found in the mouth cavity or also in terms of grains such as barley. So the maltose that is been fermented either in partially or completely by microbes will then lead to ethanol, and therefore the liquor beverages are made in this manner by converting maltose to become ethanol. Maltose also, being a bit sweet, can be used in the candies. And then maltose is a reducing sugar simply because one of its sugar, the glucose, has the hemiacetal. Of course, the other glucose is already bound with the glycosidic bond, therefore it is unable to mutarotate. But the other neighboring glucose has hemiacetal, therefore it can then mutarotate to reveal its carbonyl group, and hence it's a reducing sugar on a whole as maltose. So just like maltose, lactose is also a reducing sugar because one of its member is hemiacetal and therefore can mutarotate to reveal the carbonyl group. In terms of lactose, it is made up of glucose and galactose, unlike maltose, which is glucose and glucose. And the bond that is keeping together this disaccharide is beta 1,4 glycosidic bond. I hope you understand what's the difference between the alpha and beta 1,4 glycosidic bond. It's based on the position of the OH, whether the OH is upwards, and therefore that's beta. If it's positioned downwards, therefore it's alpha 1,4 glycosidic bond. And lactose is mostly found in milk, being 4 to 5% of cow's milk. The other material in milk is protein. And this is the structure given for lactose in terms of having beta 1,4 glycosidic bond and made up of galactose as well as glucose. In normal individuals, the hydrolysis of lactose will cause the yield of galactose as well as glucose. This galactose and glucose can then go through from the digestive tract to the blood system, so no problem. But some individuals do not have the enzyme lactase sufficiently, or lacking compared to the majority of individuals. When these individuals consume milk, it will build up in the digestive tract and cause certain symptoms such as abdominal cramps, pain in the stomach area, nausea, feeling dizzy, wanting to faint or even faint itself, and then have diarrhea issues. So to basically overcome these, it's possible to take lactase, the enzyme, as supplement in form of capsule or tablet before actually consuming fresh milk. So now we move our attention to sucrose. Sucrose is also known as table sugar because it is possible to extract from beetroot or sugarcane, which is more in our country, to become table sugar, and this is what we use in everyday or daily life. But in terms of bigger production done in a factory, it's possible to also do sugar in the form of aspartame, that is entirely not carbohydrate. This one I already explained to you in the lecture of protein. So sucrose is made up of fructose as well as glucose. Unfortunately, the link that is keeping together sucrose is due to alpha beta 1,2 glycosidic bond, and therefore none of the hemiacetal, which normally consists in the individual monosaccharides, are free. They are actually to be bonded and secured. Therefore, the whole sucrose molecule is not considered reducing sugar because of the fact that no hemiacetal is available. But if can hydrolyze the bond, the 1,2 glycosidic bond, to release glucose as well as fructose, then they can become reducing sugar, but not sucrose as a whole. So here shown the hydrolysis of sucrose, where in the small intestinal tract there is the enzyme of sucrase, which can cleave the 1,2 glycosidic bond to release glucose as well as fructose, and these two monosaccharides then can be diffusing from the digestive tract into the bloodstream. So no problem in digesting sucrose if sucrase is present in the digestive tract. We move our attention to the polysaccharides. We start off with starch. Starch is found in plants, and here shown the amyloplas, where it is found in the plant leaves, and it has storage of starch. Starch is made up of two components, 20% of amylose as well as 80%, around 80% of amylopectin. Amylose is unbranched. It is helical in structure, while amylopectin is having branches of 1,6 glycosidic bond, and shown there the diagram for amylose as well as amylopectin. More information on starch and its components. So the amylose will be comprising around 20%, and it has only alpha 1,4 glycosidic bond, and it is in helical arrangement. It can actually trap iodine, therefore iodine is an indicator of the presence of starch. While amylopectin is around 80%, and it has both alpha 1,4 glycosidic bond as well as the branching bond of 1,6 glycosidic linkage. And since both amylose and amylopectin are water soluble, therefore they as a whole, starch is also water soluble. And there is necessary enzymes in the human digestive system, that is alpha glucosidase. Therefore, it is possible to utilize starch by humans itself. Here shown the helical structure of amylose as well as the trapping of iodine, and once it's trapped it will change color to blue, and that is the indicator of the presence of starch. Next, we move our attention to glycogen. Glycogen, unlike starch, is more of a storage macromolecule in animals. And in fact, they are stored in liver as well as muscle for ease of obtaining glucose, because glycogen stored at liver and muscle will be immediately broken down to glucose, and therefore glucose is the one oxidized in these organs to become ATP. And in terms of the structure of glycogen, it can be differentiated with amylopectin to be having more branching than that of amylopectin. And in terms of hydrolysis, we in our body we have all these enzyme alpha beta amylase as well as most importantly glycogen phosphorylase to convert glycogen to become into become eventually glucose or free glucose, which can then be oxidized to become ATP. Here shown some diagrams to differentiate between amylopectin as well as glycogen. You can see more branching in glycogen compared to amylopectin that is found in starch. And also on top, the diagram shown the hepatic cell or hepatocyte, liver cells having concentrated storage of glycogen, and it is freely available for converting to glucose and can be used by liver itself for the purpose of oxidation to carbon dioxide and water as well as energy in ATP. Then we move our attention to cellulose being a polysaccharide. Unfortunately, humans do not have the enzyme required. The enzyme is beta glucosidase. Uh, since glucose is the foundation in building cellulose, many glucose is required into making cellulose. But the type of glucose is beta glucose rather than alpha glucose, and therefore the bond that is formed in cellulose is beta glycosidic bond, and this one cannot be cleaved by alpha glucosidase that is found, the enzyme found in your body, but the animals, ruminants such as cows, deer, giraffes, camels, they have many stomachs, and within their stomach got microbes with beta glucosidase, and they can digest cellulose found in grass, and therefore they can utilize it. This makes cellulose as the most abundant carbohydrate because it cannot be literally consumed easily. Here shown the difference between starch component as well as cellulose component. You can see the starch is made of alpha 1,4 glycosidic bond, uh, while compared to cellulose made up of beta 1,4 glycosidic bond. So that's the reason the inability to digest cellulose in comparison to starch. Interestingly, here shown the microfibril of cellulose, where it is made up of a long chain of this glucose chain made up of 1,4 glycosidic bond It is the microfibril is brought together. The chain is kept together by hydrogen bond, and finally the microfibril will be used in building the cell wall of leaf, and that's how cellulose is strong. So once we have covered monosaccharide, disaccharide, polysaccharide, we now move our attention to modified carbohydrates by starting with chitin. So chitin is unlike a chain of glucose, it is having the modified monosaccharide, that is glucose that has been removed of its OH and attached with amino as well as acetyl group. So therefore it is referred as the N stands for where the nitrogen connect to the sugar, and then acetyl glucose amine. So this is the monomer for chitin, and this chitin is unbranched, and it is it is found in the exoskeleton of the arthropod or insect. So more sugar derivatives been shown here. You have the ribitol comes about because of the glucose remove of the carbonyl group and attached with CH2OH. So it's alcohol. So it's known as ribitol. Then you have the glucose, remove the carbonyl group and attach with carboxylic group. Therefore, it is referred as gluconic acid. And also, at the bottom of the acyclic glucose, instead of CH2OH attached with carboxylic group, therefore, it is now referred as D-glucouronic acid. So here given glucosamine as well as acetyl glucosamine, and it is based on the OH of glucose being replaced with amino group, therefore it's glucosamine. Then you have the OH replaced with amino as well as acetyl, therefore it is known as acetyl glucosamine. And here an example on a very complex sugar derivative, and it is known as acetyl neuraminate or sialic acid, and it plays a role in glycoprotein where the glyco part will be the sialic acid, and it gives a negative charge for the glycoprotein. Glycoprotein, the protein part will be at the cell membrane, on the membrane itself running transverse or inside the cell membrane, while the glyco part will be outside of the cell membrane, outside of the cell. And this sialic acid, apart from the carboxylic group, it also has the N-acetylate and also the R group here, some complex structure given, and it is known as sialic acid or acetyl neuraminate. So next we look at conjugates. That means the carbohydrate conjugated with protein. There are two, proteoglycan, glycoprotein, and also the carbohydrate conjugated with lipid, that is glycolipid. The proteoglycan is found in the space outside of the few cells or outside of the cells, and this space is referred as extracellular matrix or ECM, and they proteoglycans having these repeats known as glycoaminoglycans, while glycoproteins are attachment of protein with a few oligosaccharides, and glycolipids are found in the membrane lipids, where the lipid part will be in the cell membrane or membrane, and the glyco part will be on the outside, having certain functions. So here shown the difference between glycoprotein as well as proteoglycan. Glycoprotein does not have the serial repeat unit of glycoaminoglycan, and compared to proteoglycan, this one has more protein rather than carbohydrate. And in terms of proteoglycan, they have these serial repeats that is referred as glycoaminoglycan, or in abbreviated as GAG. And they have proteoglycan, more carbohydrate, less of protein. So here shown the proteoglycan, where it has serial repeats of glycoaminoglycan. Here you can see, and n determines how long it is. This whole structure will be outside of the cell, while the protein part will be within the cell membrane. And this structure, which is outside of the cell, plays a role having many functions, such as being a lubricant. Therefore, mechanically, the cells are not damaged. And also, it plays a role in simulating cell proliferation. That means cell growth after the cell has been formed, among other functions. So here given the modified sugar being attached to amino acid, three types shown here. You have type 1, you have the modified sugar N-acetylated glucose attached to asparagine. Then you have N-acetylate amino glucose attached to serine or either threonine. Then type 3 glucose, which is attached to modified lysine. So from this point onwards, there will be a lot of this glycoaminoglycan being referred. It gets very complicated, but do not worry, you do not need to memorize them or know them in detail. Just know their presence or existence. Here given hyaluronate, it is made up of glucouronic acid because of the carboxylic group. Then you have N-acetylglucoamine group because of this. And within the repeats, it's done by beta 1,4 glycosidic bond. And between the sugar derivative, it is beta 1,3 glycosidic bond. So next you have chondroitin sulfate, where it is made up of glucouronic acid and acetyl galactoamino group. And the connections within these two monosaccharide or derivative is beta 1,3, and the repeats done by beta 1,4 for chondroitin sulfate. Additionally, it also has the sulfur group there to signify its chondroitin sulfate. So now you have more of the GAG. This one is heparin as well as heparin sulfate. You can see the difference between both of them. I'm not going to go in detail on each one of them because for sure I'm not going to cover them in assessment. You just need to know their existence for sugar derivatives. So as you can see, But I also want to emphasize all these glycoaminoglycans, such as heparin, have a certain function. Heparin, for instance, can serve as an anticoagulant. They will put into vacutainers, tubes that they collect blood. They will coat it with heparin so that it does not coagulate, and therefore it will be still in liquid form. So this covers in terms of keratin sulfate 1, also keratin sulfate 2. Not going to go in detail, but this is how it looks like in terms of its structure. Finally, we come to this slide here. It's about why in nature the protein, once it's undergone transcription process and then translation, then you get a protein. Why it undergoes certain post-translational modification, which adds carbohydrate to the protein. So in nature, protein does not really only solely become protein. It actually undergoes modification to add on purpose carbohydrate. So what is the function of the carbohydrate for the protein, the conjugate? So these are all the reason been identified. The protein is conjugated to the carbohydrate. The first one is the protein, if it is made up of hydrophobic amino acid, by adding carbohydrate, it is actually better in terms of solubility to water, because carbohydrates contain a lot of OH group. Therefore, the whole protein become polar, soluble in water. Next, the folding. It seems the protein, once it become polypeptide chain, and then it starts to bend and fold, obviously because of the R group, and having the carbohydrate included, it will tend to fold to a particular 3D conformation, which is specific for that protein, and hence the whole functionality of the protein. So that carbohydrate determines the structure and therefore the function of the glycoprotein. The third facts is having carbohydrate. Microbes have protease enzyme. So microbes can degrade or spoil, for instance, spoil food when you leave it out in the open. So the protein which has the carbohydrate included will deter protease to degrade the particular protein, and therefore the carbohydrate protects the protein from being damaged and spoiled. And lastly, since carbohydrate is part of glycoprotein, hence studying the carbohydrate will reveal a lot of information about the functionality of the carbohydrate, the glycoprotein itself. And this is the last slide. In the usual manner, we end by having a student activity. In total, it's not easy to understand this carbohydrate lecture simply because it involves a lot of chemistry terms. Not all you need to actually memorize, only to understand, and also you have to understand that the lecturer cannot assess every single part of this carbohydrate lecture. So please be selective in learning, and do not give up in learning just because it seems more complicated than other lecture.